Well, good morning, good afternoon, everyone. First of all, I'd like to thank the Royal College of Pathologists and the Ghana College of Physicians and Surgeons for this kind invitation. The topic of my presentation will be gene therapy for sickle cell disease. Just a few words about myself as an introduction. I am Emmauel Payen. I am a researcher for the French Institute of Health and Medical Research. I've been working for about 20 years to develop gene therapy programs for hemoglobin disorders. First at the Institute of Hematology of the Saint-Louis Hospital in Paris with Professor Yves Bezard, a specialist in hemoglobin disorders, then at the site of the French Atomic Energy Commission with Professor Philippe Lebouche, a specialist in lentiviral vectors. I was also a consultant for genetics pharmaceuticals, which later became Brood Bio, with whom we developed a lentiviral vector for beta thalassemia and sickle cell disease. During this presentation, I will give a very brief introduction to sickle cell disease and the value of gene therapy. I'll spend some time to discuss the methods being considered for sickle cell disease, most of which are also valid for beta thalassemia. I'll talk about clinical successes. I'll discuss the potential risk and how they are being addressed. And I will conclude by indicating the status of the gene therapy product that I will have described to you, either in clinical trials or awaiting a marketing decision following the filing of a biologics license agreement application. As you know, sickle cell disease is the most common genetic disease in the world, with more than 300,000 births worldwide per year, and a significant increase is expected in the coming years due to the decrease in children mortality. The majority of children with sickle cell disease are born in Africa, and almost half of them die before the age of 10. India is the second most affected part of the world by the disease. In high-income countries, patients mostly pass the childhood stage and live up to 50 years of age or even older. This very large disparity is related to many factors, including genetic screening, early diagnosis, education of both caregiver and parents, and of course access to care, including vaccination, prophylactic therapies, use of hydroxyurea, access to blood transfusion, and pen treatments. In countries with sufficient financial resources, as well as sophisticated hospital and infrastructures, allogeneic transplantation is currently recommended for all severe sickle cell patients if they have a geno-identical intrafamilial donor. This so far applies only to the severe patients, which are those who have had a stroke or are likely to have one, those who suffer from frequent painful vaso-occlusive attacks and recurrent episodes of acute chest syndrome. In this case, the benefit are considered to outweigh the risk of mortality, graft versus host disease, infections, or graft failure. Well, an alternative to allogenic bone marrow transplantation is gene therapy. Unlike in the case of allogenic bone marrow transplantation, where cells were obtained from an NHEJ identical intrafamilial donor, gene therapy involves the patient's own cells that are genetically modified ex vivo before they are injected into the patient. As with allogenic transplantation, the cells are injected after myeloablative conditioning. The difficulty of gene therapy is twofold. First, the rate of modification that must be as high as possible to come close to the efficacy of allogenic transplantation. And second, the effect on stem cell must be non-toxic, both in the short and in the long term. Gene correction of autologous cells has quite some advantage over allogenic transplantation because it avoids not only the risk of rejection by the recipient's immune cells, but also, and most importantly, the main cause of morbidity and mortality that affect patients who are transplanted with allogenic cells, which is graft versus host disease and all the risk associated with this disease. Another very important issue with allogenic bone marrow transplantation is the fact that less than 20% of sickle cell patients have an available intrafamilial donor. Therefore, In theory at least, if gene therapy effectiveness is recognized as well established, gene therapy should allow to treat 100% of patients eligible for myeloablative treatment. Obviously, it's not black and white. There are also potential risks that are inherent to hematopoietic stem cell gene therapy. The risk associated with the myeloablation procedure, which is the same as for allogeneic transplantation, since the recipient cell must be eradicated anyway. It may even be a little bit higher because it seems that the myeloablation must be as deep as possible, and therefore the dose of the myeloablative agent must be maximized for gene therapy. There is a genotoxic risk, which is due to the manipulation of the genome, whether by the insertion of viral vector or by the use of meganucleases, a risk, of course, that does not exist for allogeneic transplantation. Finally, there may be risk with regard to the quality of the injected stem cell, or as they are manipulated in vitro and grown for some time, a few days, before they are injected back, and therefore the long-term persistence may change as compared to the fresh cells that are injected during allogeneic bone marrow transplantation. There is therefore a clear rationale, first, to increase the number of patients who can have access to marrow transplantation, and second, to reduce the immune risk, but there are also additional risks inherent to gene therapy that, of course, need to be measured and evaluated. Well, the question then arises as to which hemoglobin is the best to replace sickle hemoglobin in order to treat sickle cell patients with gene therapy. Would it be more effective to express beta or gamma globin chain? Well, the answer, as you probably know, is of course gamma globin, in order to produce fetal hemoglobin, given its widely demonstrated impact on disease severity. A high level of fetal hemoglobin has been shown to improve the survival, the risk of stroke, pain, occurrence of acute chest syndrome, leg ulcer, etc. So clearly, it is preferable to express the gamma globin chain of fetal hemoglobin rather than the beta globin chain of adult hemoglobin. Well, the reason for the advantage of fetal hemoglobin over adult hemoglobin is quite simple to understand. For aggregation of hemoglobin S tetramer to occur, the tetramer needs two things: a valine in position 6 and a hydrophobic pocket, hydrophobic valine accepting pocket in position 85 to 88, consisting of a phenylalanine, a threonine, and a leucine. In the non-mutated beta A chain, there is no valine, but still the hydrophobic pocket is present here. Thus, the heterodimer, made of one beta S and one beta A, can perfectly participate in the polymer formation as shown here. On the contrary, the gamma chain of fetal hemoglobin has neither the valine nor the hydrophobic pocket, and therefore the heterodimer cannot participate in the polymer formation. Under this condition, the beta S chain included in the heterodimers are excluded from the polymer formation. Therefore, not only HbF prevent polymerization by lowering sickle hemoglobin concentration, but also the presence of gamma globin prevents the beta S chains from participating in the polymer formation. This is the reason for which it is preferable to express a gamma globin chain than the beta A globin chain to prevent polymer formation in patients with sickle cell disease. So basically, there are two main strategies for gene therapy, here shown in these two red squares. One on the left is to dilute sickle hemoglobin and to inhibit cycling. The second, that I did not discuss yet and which may be more complicated, is to repair the beta S mutation, the valine at position 6. For each strategies, there are several technical possibilities. The first technique, the most advanced and the oldest one, is gene addition using lentiviral vector. The vectors can express a cycle cell inhibitor, genes such as the gamma gene or a chimeric beta-gamma globin chain that does not contain the valine at position 6, of course, and that does not contain the hydrophobic acceptor pocket. The lentiviral vector can also express an inhibitor of BCL11A, for example, an shRNA targeting BCL11A. Why BCL11A? This is because this protein is one of the master regulators of gamma globin gene expression. In erythroid cell, it is expressed and it inhibits gamma globin gene expression. So by inhibiting the expression of BCL11A, it is possible to re-express the gamma globin genes in adult erythroid cells. The second technology, which is much more recent, is genome editing. This technique can be used to either reactivate gene expression or to repair the genome. In one case here, the technique can be used to directly correct the causative mutation of sickle cell disease and therefore the BILA-S gene. In another case here, it consists of reactivating the expression of the gamma globin gene. And there are basically two options. One is to use the editing system to disrupt BCL11A expression in erythroid cells, and that is again to stop the inhibition of the gamma globin gene expression in adult cells. Another possibility is to edit the genome close to the promoter region of the gamma globin gene in order to mimic HPFH mutation, which are known to reactivate gamma globin gene expression. Well, we'll speak about all of these possibilities in the next slides. Let's first have a look on lentiviral vectors which derive from HIV-1. The basic principle to make a vector from a virus is actually very simple. It is a matter of eliminating all the HIV genes that are responsible for the replication of the virus and keeping only the cis-acting elements here that are strictly necessary for the retrotranscription of the RNA genome into double-strand DNA and the integration of the viral DNA into the genomic DNA of the target cells. When the vector is ready, the objective is to maximize the transduction efficiency in order to obtain the highest proportion of transduced cell as possible. Then the vector and the transgene are stably integrated in the DNA so that the cell progeny, including the erythroid cells, which are differentiated from the stem cell, will also contain the transgene. If one has introduced an erythroid promoter here to control the transgene, for example, gamma globin gene, then the gene will be expressed specifically in erythroid cells. The small difficulty is to produce lentiviral-like particle if the vector does not contain any viral gene. The principle is described on this slide and is based on the use of packaging cell lines into which you need to introduce several plasmid DNA, the vector plasmid DNA, of course, that encode the RNA genome that must be packaged, and all plasmid DNA containing genes that express the protein which are required for production of the viral particles. When all these plasmids are transfected this way, the packaging cells then produce a functional vector associating the RNA genome that contains the transgene, but also all the protein which are required to encapsulate the RNA genome into a viral-like particle that will then be able to enter the target cells and to deliver its genome into the cells. In case of hematopoietic stem cell, the envelope that is used is the one derived from the vesicular stomatitis virus, the VSVG envelope, the receptor of which is the LDL receptor. So the particle first enters the cell by endocytosis, and then the RNA genome is reverse transcribed in double-strand DNA by a reverse transcriptase. The reverse transcriptase has been incorporated in the viral particle. So it is reverse transcribed in double-strand DNA that is then transported inside the nucleus, where the integrase that is also transported by the viral particle allows integration of the double-strand DNA into the genome. The most important advantage of this method is the efficiency that can be quite high, with recent protocol improvement that have shown efficacy close to 90–100%. Importantly, and it was a concern many years ago, but it's not the case anymore, the production method in packaging cell precludes the production of replication-competent lentiviruses. No replication-competent lentiviruses have been detected so far in production lots. The disadvantage, as you may know, of the use of lentiviral vectors is their semi-random integration in the genome, and thus, of course, the potential risk associated with insertional mutagenesis. But this is something that we will discuss a bit later. The second great series of tools dedicated to gene therapy, and in particular to genome editing, the one that has revolutionized the world of biotechnology in the last decade and now gene therapy, comes from the discovery of the system that is well known as the CRISPR-Cas system. This tool, in fact, is derived from the bacterial immune system that bacteria have developed to protect themselves against foreign DNA, either plasmids or bacteriophage. During an infection, for example, bits of foreign DNA are integrated into a locus, a bacterial locus, that is called the CRISPR locus. Then this bit of DNA can undergo a transcription to form CRISPR RNAs. Together with the Cas nuclease, they form a complex that scans foreign DNA for the presence of the target sequence. And when they encounter a compatible sequence, so a homologous sequence, the nuclease, the Cas9 nuclease here, snip the DNA at a very specific location within the target sequence. This is the way how bacteria can fight against virus and plasmids. Jennifer Doudna and Emmauel Charpentier were the first in 2012 to propose that this system can be adapted and used as a programmable toolkit for genome editing in eukaryote. This system, in fact, is pretty simple, as it only requires the presence in the cells of two components. First, the Cas9 protein, the nuclease, which is made of two domains, the RUVC and NHEJ domains, each domain capable of cutting one DNA strand, and made as well of the guide RNA. The guide RNA is a short synthetic RNA that is made of a scaffold sequence, here in orange, which is necessary for Cas binding, and 20 nucleotide spacers that define the genomic target to be cut by the Cas nuclease. The sequence that is recognized by the complex is cut only if there is sufficient homology between the guide RNA and the target sequence. It is therefore a very versatile tool, since it is enough to change these 20 nucleotides here in the RNA sequence to modify one gene or another. The only constraint is to meet two conditions. The first is that this 20 nucleotide motif is unique compared to the rest of the genome, otherwise the nuclease would cut at several positions, and that this 20 nucleotide motif is followed by a small motif of a few nucleotides that is called the PAM, for protospacer adjacent motif, the presence of which is necessary for the Cas nuclease to bind DNA. For Cas9, for example, the PAM sequence is NHEJ, but there are other Cas and Cas9 variants with different motifs if required. So what does the CRISPR-Cas system when it recognizes a specific DNA sequence? In fact, thanks to this two nuclease domain, it cuts the DNA on the two strands and thus makes what is called a double-strand DNA break. Once a double-strand DNA break has been generated, what are the options for cell? When this happens, the DNA break can be repaired by two distinct pathways, either the error-prone non-homologous end-joining pathway, abbreviated as NHEJ, or the precise homology-directed repair pathway, abbreviated as HDR. The NHEJ system repairs double-strand break by ligating the free DNA ends, but most of the time it also introduces deletions or specific sequence at the joining ends, thus creating what are referred to as indel mutations. Thus, in this case, if a gene is targeted, it will be disrupted. On the contrary, in the presence of a DNA template, the homology-directed repair pathway follows a directed correction strategy through a recombination with homologous adjacent sequence. In this case, the break is exactly repair. What is interesting is that if the DNA template, like this one here, is slightly different from the broken DNA, here with the green nucleotide, for example, some new DNA information can be incorporated in the corrected DNA. Of course, it's also possible to delete DNA sequences. The homology-directed repair is obviously advantageous when the objective is to precisely correct a mutation. But the choice between the two pathways for the cells is not straightforward, and in fact it depends on many factors, including the presence and the concentration of the DNA donor template, the length of the homology arms of the donor DNA, and most importantly, the relative activities of the repair system in the cells. It depends on the cells, in fact, and it depends mostly on the cell cycle. In mammalian cells, the NHEJ pathway is predominant. Furthermore, HDR works only during late S and G2 phases of the cell cycle, whereas NHEJ worked at any time. Thus, if cells are in G0 or G1 phases of the cell cycle, as it is the case for Excessiles, the rate of homology-directed repair is quite weak as compared to NHEJ. Another problem is the activation of p53 by the introduction of DNA double-strand break, which is further increased in the presence of double-strand DNA, which may generate a lot of cell toxicity. The addition of all these difficulties induced that the frequency of HDR-corrected target, even in the presence of a DNA template, is never higher than 50% in any cell type or any locus. And it's even worse in long-term hematopoietic stem cells due to the fact that they're mostly quiescent. So in fact, in those cells, even in the presence of DNA template, at most target site, the frequency of indel due to NHEJ is pretty high. Anyway, the CRISPR-Cas tool is really interesting because it is a modular and programmable system. It is possible to cut at the desired position in the genome, either to induce a cut and generate a series of cells with small insertion or deletion, which will invalidate the gene, or maybe in the presence of a template to correct a mutation by introducing a new DNA fragment. Well, this is the theory, because in the reality, it is a little more complicated than that. As one can imagine, there can be a cut at other sites than the desired location, which is called off-target, and also there can be unexpected event at the desired site itself, which is then called on-target genotoxicity. Whereas off-target events have been well described, are quite predictable based on sequence homology within genomes, and the problem has been largely resolved by improving the fidelity of Cas protein. On-target genotoxic events are probably still underestimated. Well, let's start with off-target genotoxicity. This can happen because the Cas9 nuclease can cut DNA even if the homology is not perfect, and especially if imperfections happens within 10 bases 5' of the guide RNA. There are basically two situations. If the on and off sites are on different chromosomes, the generation of a double-strand break at the off site can induce the invalidation of a gene, but it can also induce chromosomal translocation between the two chromosomes targeted by the nuclease. Also, if the sites are on the same chromosome, I mean the on and the off sites, then cleavage at both sides can result in deletion or chromosomal inversion of several kilobases or several megabases. The problem with Cas9 is that this phenomena had been observed with a frequency of a few percent under certain conditions and some location, which is obviously far too high. This is the reason why this issue has come under scrutiny. An important thing is the fact that there are a large number of tools and software that have been developed for minimizing and detecting off-target site in order to evaluate the risk of off-target investigation. Something that has become clear also is the fact that the Cas9 protein must be present in the cells at a limited concentration and for a limited amount of time in order to limit its presence to the strict requested time, so that the method used to deliver the protein, which must be transient, is really important. And finally, as already stated in the previous slide, protection against Cas9 cleavage at off-target site have been developed, especially by using Cas nuclease with enhanced specificity such as Cas1 Cas9 or Cas9 HF1. It is likely that non-specific split still exists, but it has been considerably reduced and is now considered almost resolved. Well, while off-target genotoxicity is almost part of a past issue, and today part of the evaluation following the use of the CRISPR-Cas technology in ongoing gene clinical trials, the awareness of on-target genotoxicity is more recent. On-target genotoxicity concerns every kind of unwanted rearrangement that may happen at the target site. Most of the time, as I just described, indel mutation or below 50 base pairs, so that they only affect the target gene and even a specific site within the target gene, which can be, for example, a regulatory DNA element. But sometimes, and that has been observed in many cell types and at several loci, much larger rearrangements can happen. This large-scale modification includes interstitial insertion or deletion, interstitial copy-neutral loss of heterozygosity, terminal deletion, terminal copy-neutral loss of heterozygosity, or even more catastrophic events such as chromotripsis and chromosomal loss. Retrotransposon insertion events has also been observed. Most of these large-scale rearrangements, however, have been observed in immortalized cell lines, as they mostly occur in the absence of p53, which is obviously present in primary cells. Nevertheless, copy-neutral loss of heterozygosity, also called uniparental disomy, has been observed at a frequency of up to 1% in hematopoietic progenitor cells. This kind of loss of heterozygosity and duplication of one of the parental chromosome is a genetic event that has been shown in various cancers, including hematologic malignancy. The mechanism by which they may contribute to tumorigenesis is by duplicating a mutated oncogene or by deleting a functional copy of a tumor suppressor gene. Of course, this danger must be put into perspective for the moment, since to date, the patients included in clinical trials in which a double-strand break was induced by the CRISPR-Cas9 system have not developed leukemia. The most likely reason for that is that the severe adverse events that compromise genomic integrity typically result in cell death due to p53 activation. However, and that is the most important risk factor, the risk of a selective advantage and proliferation given to some of the cell is possible, especially for those of the cells lacking p53 if they exist in the pool of a patient cells. This issue, for sure, will require further investigation and is a risk factor that has to be taken into account when including a patient in such a protocol. So there is a risk. It is still theoretical, but an existing risk in using the CRISPR-Cas system to generate double-strand break as a gene therapy tool. In order to reduce this risk of on and off-target mutagenesis, an idea has emerged, which is to use the CRISPR-Cas system not to make a double-strand break, but and invalidate a gene or provide a DNA template, but to provide a protein whose activity would be to modify a DNA sequence. Thanks to this idea, two technologies were born, namely base editing and prime editing. In the case of base editing, we still have the two basic components of the CRISPR-Cas system, which are the Cas protein in gray and the guide RNA to target a specific sequence in the genome. But in this particular case, another protein domain, cytidine deaminase, is fused to Cas9 in order to convert cytidine into uridine within nucleotide 4 to 8 of the protospacer, leading to a UG mismatch. In principle, in a cell, the UG base pair should be recognized as a mismatch by the cellular repair machinery that would normally remove the uracil. The cellular enzyme required for this U here to be removed is the uracil DNA glycosylase. For this reason, an extra protein domain, an uracil glycosylase inhibitor, UGI, has been added to Cas9 in order to prevent the reversion of U in C. Another modification of the system is the mutation of Cas9, the D10A mutation, so that only one of its two nuclease domain, the HNH nuclease, cleaves only the complementary strand, which is the non-edited strand upstream from the PAM sequence. This nicking step is quite important because it will favor the use of the U here as a template. The use of the U on the non-nicking strand to replace the G in the A, as shown here by the DNA mismatch repair machinery. Thus, after repair and replication, the GC base pair will have been converted into an AT base pair. A very similar system has been realized in order to convert AT into GC. I'm not going to describe that into much detail, but basically the principle is quite the same. The first system that converts G in A and C in T is called a cytosine base editor, or CBER, and the second system that converts T in C and A in G is called an adenine base editor, or ABE. The scope of base editing system is limited to 6 kinds of nucleotide conversion between purine here, A to G and G to E, between pyrimidine, C to T and T to C, and also, I did not talk about that, but it's also possible to convert G to C and C to C. So all of the possible combinations are shown here. But as you can see, most base transversion are not possible, especially the conversion of a thymine into an adenine, as it should be done to correct sickle cell disease. Finally, and I will stop here for the description of this tool, this CRISPR-Cas derived tools. Another approach has been described in 2019 by the group of David Liu in Cambridge, and this a very interesting approach has the potential to write any kind of genetic information into a specified DNA site. This system can be used to perform small insertion, small deletion, and all kinds of point mutation. Well, it still relies on the CRISPR-Cas system, but this time the Cas9 protein is fused to reverse transcriptase. The CRISPR-Cas9 protein is mutated to become a nickase that will cut the PAM strand only. That time is a bit different from the previous system. Another important element in this system is the guide RNA, of course, that is still necessary to recognize the target sequence, but in this system, this guide RNA is also fused to what is called a prime editing RNA. This incorporated prime editing RNA sequence is made of a primary binding site and a template for the reverse transcriptase. I'm not going to describe the mechanism in detail, it would be too long, but at the end, this system results in the production of two kinds of DNA, either the non-edited wild-type DNA or permanent modification that can be deletion, insertion, or base conversion of the target DNA sequence. I have tried here to summarize the property of these tools in order to compare the most important points to my opinion in the field of gene therapy. First of all, the efficacy of genetic modification. It's now very high with lentiviral vector, and after optimization, it has been shown to reach level up to 90%. It's also very high when the objective is to use CRISPR-Cas system to induce NHEJ, probably quite a bit less for base editing, and certainly still much lower for prime editing. And it's even probably less for homology mediated repair, especially in long-term hematopoietic stem cell. An advantage concerning the expression level very likely goes to the repair system, since in those cases the expression level will be physiological, whereas in the case of gene addition by lentiviral vectors, then the expression level will depend on the vector copy number and the position of the vector in the gene. Importantly, the genotoxic risk may be quite different. The risk is well established for a lentiviral vector, which is mainly the activation of oncogene, possibly. For the other system based on CRISPR-Cas technology, most of the genotoxic risk will depend on the rate of double-strand breaks. Although it is considerably reduced with the base and prime editing system compared to a system in which double-strand break is deliberately inserted, double-strand breaks still happen with prime and base editing systems, so that it may still be an issue with base and prime editing. It is therefore very likely that with the high number of hematopoietic cells that are modified and injected into the patients, some of the cells, even with this single-strand break system, will have chromosomal rearrangement. This, of course, will need to be looked very closely during ongoing and future clinical trials. Finally, a word about the cost. The production of viral vector is pretty expensive. In fact, it's difficult to produce the vectors, and quite more expensive than the production of RNA and ribonucleoprotein complexes. So in terms of cost, there is likely an advantage to non-viral technology. Of course, that will depend on the price at which the company will sell the product. That may be independent on their production price, of course. I'm now going to describe some important results obtained with lentiviral vector and the editing technology. But let's first start with clinical trial realized with lentiviral vectors. And I choose on purpose only to describe the most advanced product, which is derived from the use of this vector, the BB305 vector, this one, that we set up in our laboratory together with Bluebird Bio, and that contain a modified version of the beta-globin gene. It's a gene with the mutation at position 87, so that the threonine of the hydrophobic pocket that binds to the mutated valine is changed into glutamine. According to experiment made a long time ago in vitro, and we confirmed that this modification is thought to be sufficient to transform the beta-globin chain in a chain having the same inhibitory activity as the one of the gamma-globin chain. Several clinical trials have been conducted with this vector from 2007 in France up to now. These clinical trials have included patients with beta thalassemia and with sickle cell disease. The product have two different names according to the target population, either beta-cell for transfusion-dependent thalassemia or lovo-cell for patients with sickle cell disease. There have been a number of publications already, and the publication dealing with patients with sickle cell disease are underlined. The results have been published in 2017 for the first sickle patient included in the HGB-205 study, and in 2022 for most of the patients included in the HGB-206 study. The study for which we have the more patient and the more result is the HGB-206 study, which was initially a phase 1 trial, but that has been modified in a phase 1/2 later on. Also, the protocol has changed quite a bit with time, whereas the first two groups, the A and B group of patients had bone marrow cell harvested for collection here. The group C patient had a mobilization with plerixafor in order to get more cell to be transduced and infused. Three other important changes have been introduced. The conditioning that increased from group A to group B and C. The target concentration of busulfan was 16,000 micromole minute in group A, but 20,000 in group B and C. The transduction efficacy, which is the amount of gene modification per cell in the drug product, was optimized for the preparation of drug product for patient in group B and group C. And finally, in order to improve the quality of the collected cells before apheresis, patients in group B and C were hypertransfused from 2 months before collection. So a number of modifications to try to improve the protocol between patients of group A and patients of group B and C have been made over the time. The result for group A and B have been disclosed at several congresses, but they have been published only recently in 2023. On this graph, you see in green here the proportion of HbS, and in red the proportion of the therapeutic hemoglobin. You can see that for group A patient, the proportion of the therapeutic hemoglobin was pretty low, between 7 and 28%, whereas it was up to 35–54% for the two patients in group B. This is mainly related to the percentage of transduced cell, of course, measured by the average vector copy number per cell in the peripheral blood, which was only about 0.1 in group A patient, while it was 0.7 and even higher than 2 in one of the group B patients. From the result in this early included patient, it could therefore be concluded that the evolution of the protocol between group A and group B, the improvement of the quality of the collected cell, the higher proportion of transduced cells, and the higher intensity conditioning may have collectively led to a clear increase in the amount of transduced cells and thus the amount of the therapeutic protein. It was thus decided by the sponsor to include more patients to clearly establish the effectiveness of the treatment. As I said before, a modification in group C compared to what was done from patients in group A and B was the harvesting of the hematopoietic progenitor and stem cell from mobilized blood cell instead of bone marrow, with the clear objective to harvest and inject more cell into the patients. The target number of patients in this group of this study was 50, aged between 12 and 50, with a history of severe vaso-occlusive events not resolved with hydroxyurea. In this trial, the efficacy endpoint were the resolution of severe vaso-occlusive event, the production of modified hemoglobin at a level believed to be therapeutic, so higher or equal to 30%, and a significant increase of total hemoglobin. For the first 35 patients included, the mean number of 2 mobilization cycles were required to get the correct number of cells. The median number of injected CD34 cells varied between 3 and 25 million per kilo, and time to neutrophil and platelet engraftment after myeloablation and infusion was 20 and 36 days. You can nevertheless see that platelet recovery could be quite long for some individuals, for sure longer than expected. It raises, of course, the question of to know whether the transduction protocol may have in some cases modified the short-term recovery capability of platelets. As shown here on this slide, modification of the protocol translated into a very strong improvement in all the parameters studied compared to what had been obtained in group A patient. The number of vector copies was between 0.5 and 3 and even higher for 2 patients. The quantity of the therapeutic hemoglobin was around 5 grams per deciliter, and the proportion of the anticycling hemoglobin was greater than 40% in all patients. Vaso-occlusive crisis, or what bothers patients the most, which is why the reduction or elimination of vaso-occlusive crisis is an essential parameter. This diagram here represents the number of vaso-occlusive crises before and after treatment. While the median number of crises was around 3 to 4 per year before the treatment, no severe crisis was noted after the treatment, nor any sign of acute chest syndrome, which is likely to be a very important factor for the improvement of the quality of life of patients. Although vaso-occlusive crisis is a major issue, intensity of the hemolytic anemia is closely associated with mortality, so both part of the pathophysiology, vaso-occlusive events and hemolysis, have to be addressed. It is thus very important to observe here that increased therapeutic hemoglobin production in this slide was clearly associated with an intense reduction of all hemolysis markers. In general, the safety profiles reflected perfectly the known side effect of busulfan conditioning regimen. There has been no case of veno-occlusive liver disease, graft failure, or the presence of replication-competent lentiviruses. However, 2 patients in group A died of acute myeloid leukemia. Patient 2 first was diagnosed as myelodysplastic syndrome 3 years after lentiglobin infusion and progressed to acute myeloid leukemia that was fatal. Importantly, no lentiviral vector was detected in the blast cell, indicating that leukemia was not the result of insertional mutagenesis. Patient 1 also developed acute myeloid leukemia. In this patient, the vector was detected in blast cell, but it was inserted in gene that is not known to be involved in leukemia. Furthermore, expression studies and chromosomal rearrangement studies within 10 megabase of flanking region did not show any abnormalities. Finally, insertion in this gene has been detected in most patients of this trial without any sign of hematological abnormality. It was thus concluded that acute myeloid leukemia were not linked to insertional mutagenesis. The question then arises, was these 2 patients developed acute myeloid leukemia, while group C patient did not, and beta thalassemia patient also included in clinical trials since a period of time equivalent to group A patient did not develop leukemia. So what was or were the cause of acute myeloid leukemia in this patient? First, monosomy 7 was identified in the blast cell of these 2 patients, and monosomy 7 is a type of chromosomal abnormality that is frequently detected in neoplasms secondary to exposure to alkylating agents such as busulfan. Second, the busulfan dose achieved in these 2 patients were lower than the target dose, suggesting the existence of persistent non-ablated and just non-corrected hematopoietic stem cell in these 2 patients. Finally, patients with sickle cell disease are known to have a significantly higher rate of hematological malignancy than the general population. Therefore, malignancy were attributed to the persistence of unablated hematopoietic stem cells, the use of busulfan that may have compromised the genomic stability of hematopoietic stem cells, which may already be affected by years of chronic hypoxia, chronic inflammation, oxidative stress, high cell turnover, endothelial and vascular damage, all susceptible to increase the rate of hematological malignancy in sickle cell disease. It was also attributed to the infusion of low corrected hematopoietic stem number, leading to the need for more replication cycle of persisting hematopoietic stem cell, and finally to the preservation of a highly proliferative and inflammatory bone marrow due to poor disease corrections. All of these factors, in fact, told the sponsor that the protocol had to be changed, and that's why patients were changed the protocol from group A to group B and then to group C, with increased busulfan concentration, higher number of infused cells, and higher transduction level. And all of these improvements were expected to lower the risk of myelodysplastic syndrome and acute myeloid leukemia. One patient in Group C died of cardiac arrest 20 months after gene therapy. However, this patient was severely affected by his disease before treatment with 29 severe vaso-occlusive events before gene therapy and an important history of pulmonary hypertension and venous thrombosis. At autopsy, significant cardiac abnormalities were detected. It was therefore deduced that the patient had died from complication of his disease. In the same group, two patients, one adult first and one adolescent a bit later, were suspected of myelodysplastic syndrome after they presented with erythroid dysplasia and low level of trisomy 8. However, there were no clinical symptoms suggestive of malignancy, no blast cell, no driver mutation, no clonal dominance, no chromosomal abnormality. They had low hemoglobin level, but normal blood count otherwise. Therefore, both diagnoses were amended to anemia. Very interestingly, both patients, and they were the only one in the cohort, had 2 alpha globin gene deletion. It is thus proposed that alpha globin trait, together with robust beta globin production, may have contributed to anemia and dyserythropoiesis. Following these cases, this genotype was added to exclusion criteria for ongoing studies. An important question, of course, is the durability of the treatment, and so far it seems that the genetically modified cells are maintaining themselves. This is well observed here in the study that started a long time ago, in particularly the group A and B patient, for whom we observed the proportion of stable modified cells up to 5 years after gene therapy. It has also been seen in beta thalassemia patients who were treated with a similar procedure and a similar vector, and here we can see the maintenance of the phenotype correction up to 7 years after gene therapy. To conclude, the one-time treatment leads to normalization of laboratory parameters and, most importantly, resolution of severe vaso-occlusive event and reduction of hemolysis markers thanks to the production of anticycling hemoglobin, the level of which is higher than 40% if the vector copy number is 0.5 or higher. An important information is that individuals carrying 2 alpha globin gene deleted should not receive the treatment. However, in another trial realized in France with the same vector, the result of which have been published in 2017 and 2022, patient 1 had 1 alpha gene missing, and he has been treated successfully with a normal hemoglobin level and with no vaso-occlusive event. Several results confirm the durability of the process, group A patient and thalassemia patient. Of course, longer-term observation will be required to assess the risk of hematological malignation to confirm whether optimization of the treatment protocol in Group C compared to Group A has reduced the risk of post-transplantation hematological cancer. I did not present any other data using globin-expressing vector, but there are other trials ongoing with some specificity, either using the gamma globin gene, for example, or another modified kind of beta globin, the beta S3, and with full or reduced conditioning. I just choose to focus on the more advanced expressing lentiviral vector that I just described. Let's now go to reactivation of the major modulator of sickle cell disease, the fetal hemoglobin. The most appropriate regulator for this, as we already discussed, is transcription factor BCL11A, which is an inhibitor in adult cells, as shown here, of the expression of gamma gene. Thus, if one inhibits BCL11A, the gamma gene can be expressed in erythroid cell. A theoretical advantage of this system compared to the addition of an anticycling gene such as beta 8087Q or even the gamma globin gene is that in this case, not only are the gamma gene active, but also the expression of the adult beta globin gene is inhibited because of the inviolability of the locus control region to activate the expression of the beta. This may be an advantage and facilitate the increase in therapeutic hemoglobin proportion. Well, one of the approaches to inhibit BCL11A is to use a lentiviral vector encoding an artificial microRNA under the control of an erythroid-specific system, here similar to that used for lovo cell. In this feasibility and safety study, 7 patients have been treated. They were mobilized with plerixafor and conditioned with busulfan. The number of injected cells, the target busulfan dose, the median vector copy number, and the drug product, and the duration of neutrophil and platelet engraftment were very similar to that seen in the HBG206 group C study for lovo-cel. The vector copy number in peripheral blood cells and the total level of hemoglobin were also similar. A vector copy number between 0.4 and 1.5, and a hemoglobin concentration around 11 grams per deciliter. Importantly, the fetal hemoglobin was higher than 20%, and the proportion of F cells increased from 14% at baseline before treatment to 70% after gene therapy. The 3 patients, P2, P3, and P7 here, who received regular exchange transfusion for secondary stroke prophylaxis, did not receive red cell transfusion or had a lower number of transfusions since engraftment and still had no stroke since transplantation. Patients with vaso-occlusive events before gene therapy had no severe vaso-occlusive event after therapy. Only patient 4 who had frequent hospitalization for priapism before gene therapy had several episodes of severe priapism after gene therapy, but no more episodes from 8 to 20 months. A recent update at ASH in 2022 reported the result for 10 patients and indicated that 1 patient out of 10 failed with only a very low vector copy number. Among the others, they confirmed the fetal hemoglobin production and the F cell proportion that were strictly maintained over time, between 20 and 40% for HbF and between 50 and 80% for F cells. They also reported that 1 patient carrying a deletion of 2 alpha-globin gene was cleared of his vaso-occlusive symptom while having a lower hemoglobin level than the other patient, but importantly, he did not present a severe form of dyserythropoiesis, as was shown for the 2 patients with 2 alpha-globin gene deletion treated with lovo-cel in the 206 study. The results thus provide by this study give the validation that BCL11A can be targeted by an shRNA to lead to successful fetal hemoglobin induction. It is not clear yet whether the fetal hemoglobin level detected in the treated patient will maintain protection from cycling and prevent hemolytic complication of SCD, as it was a phase 1 study. The good news is that so far no leukemia nor deaths were observed, but of course longer follow-up will be needed to assess the long-term effect. Bluebird Bio, which wished to focus on its flagship product, the antiglobin lovo-cel, recently withdrew from this program. Nevertheless, a phase 2 multi-site trial is now open under the responsibility of the Boston Children. Another possibility to knock down BCL11A is to target a specific enhancer element in the BCL11A gene, the GATA1 binding site, in a region of the genome that specifically controls the expression of BCL11A in erythroid cell. This binding site is located in intron 2 of BCL11A. Vertex and CRISPR Therapeutics have designed and tested a CRISPR-Cas9 system to break this site so that the cell repair system NHEJ introduce indel mutation at this position. A clinical trial has started in 2018 and will include 45 patients with severe SCD. The protocol for mobilization and myeloablation using a high target dose of busulfan is similar to that used for lovo-cel from Bluebird. The first result published in 2021 were already convincing, with patients having more than 40% fetal hemoglobin, a proportion that was apparently stable up to almost 2 years after injection, at least in the first patient. The most recent data reported in 2022 have shown that the 31 patients who had been injected so far were free of vaso-occlusive crisis. That has to be comparable, of course, to the number of severe events between 2 and 9 per year during the 2 years before injection. These 31 patients included 11 who had been free of vaso-occlusive crisis for at least 12 months after the last red cell infusion. The mean hemoglobin level was 11 grams per deciliter, and the mean fetal hemoglobin proportion was 40%. Most adverse events were considered to be correlated with busulfan conditioning. The time for engraftment were similar to that seen for the antiglobin BB305 trial, and the percentage of BCL11A edited allele was around 87%, suggesting a very high efficiency of the editing system in long-term hematopoietics. In conclusion, for Excessile, which is a CRISPR-Cas9 product inducing double-strand break, the one-time treatment leads to near normalization of total hemoglobin level and proportion of fetal hemoglobin comparable to what was obtained for lovo-cel from Bluebird Bio. The resolution of vaso-occlusive events is also on track. The result concerning hemolysis markers have not yet been disclosed. It will be important to know them, given the importance of this parameter on mortality, but the high level of edited cells make me believe that it will be as good as for lovo-cel. No hematological malignancy and no death has been observed. Of course, unintended off-target editing and on-target byproducts will have to receive considerable attention. There is another clinical trial ongoing, including 45 patients with transfusion-dependent beta thalassemia, and the results are also of major interest, with 42 out of 44 patients who stopped transfusion. And as for sickle cell disease, no death and no malignancy. There are two other small phase 3 studies that will have started in 2022 that will include young children with TDT and SCD, and all of the 114 patients will be enrolled in long-term follow-up study to evaluate the rate of malignancy, hematological disorders, and mortality. The zinc finger nuclease is a bit different system, different than CRISPR-Cas, of course, but they were amongst the first genome editing tools applying hematopoietic stem cell. They were made of zinc finger DNA binding domain that specifically recognized DNA, fused with an endonuclease domain of the FOC1 restriction enzyme. The binding to a specific locus, as for CRISPR-Cas9, result in the generation of a double-strand break and the subsequent activation of endogenous DNA repair mechanism. Sangamo and Sanofi have started a clinical study in 2018, the PRECISIN study, which should include 8 patients with severe sickle cell disease from 18 to 40 years. As for most of the other studies, the cell collection is made upon plerixafor injection and conditioning is performed using busulfan. First, let's discuss the percentage of edited cells. Quite surprisingly, the percentage of edited alleles measured by the proportion of BCL11A alleles carrying indel mutation fell sharply between what was measured in the injected product and what was measured in the patient cells, suggesting a relatively low editing efficiency of a long-term stem cell. For unknown reason, this is quite different to what was observed with Excessile, the product from CRISPR Therapeutics, and the reason is unknown. Despite this, quite a distinct proportion of fetal hemoglobin could be measured, of the order of 30 to 40% in at least 3 patients, and less in the 4th subject. Furthermore, the proportion of F cells were pretty high, around 90% in the 3 patients with the highest fetal hemoglobin level. Interestingly, the number of vaso-occlusive crises were considerably reduced, with only 1 event here in the patient with the lower level of fetal hemoglobin, but no events in patients with 30% fetal hemoglobin or higher. Unfortunately, no information was given on the hemolysis marker, but in my opinion, there is a risk that the correction will be much worse than with lovo or Excessile because of the large number of uncorrected, unedited cells that will most likely disappear by hemolysis. It is thus difficult to conclude on the therapeutic value of this product, even though decent level of fetal hemoglobin were detected and strong reduction of vaso-occlusive events were observed as well. Another possibility to reactivate fetal hemoglobin is not to play with expression of BCL11A, but to modify DNA elements in the gamma globin promoter region, which are known to reactivate gamma globin gene expression. There are different mutations. There are mutations such as the one shown in blue here that are known to disrupt the binding site for transcriptional repressors such as LRF or BCL11A. And there are also mutations such as the one shown in brown here that are known to create sites for transcriptional activator, for example, CALF1, TAL1, or GATA1. So there are basically 2 possibilities. Either generate an indel mutation by using the CRISPR-Cas nuclease and relying on NHEJ repair mechanism in order to mutate this repressor binding site, or change a base using the CRISPR-Cas derived adenine base editor system that can change a T into a C or A into a G. This is what Editas, the company, has chosen to develop, an editing system based on a slightly different nuclease than Cas9 called Cas12A, derived from another bacterium. The objective of their protocol is to generate indel mutation and disrupt the motif responsible for BCL11A binding site within the two gamma globin promoters. According to them, the Cas12a protein has two advantages over Cas9. First, it introduces larger deletion and thus has a higher impact on the ability of BCL11A to bind the motif and thus a higher level of fetal hemoglobin production per cell. And second, it generates a higher frequency of genetic mutation. A clinical trial called RUBY has recently started and should include 20 patients between 18 and 50. No results have been published in any journal or abstract yet, but a press release was issued by the company in December last year, giving encouraging results, with 2 patients treated and the first patient already showing over 40% fetal hemoglobin after 5 months. The results will of course have to be confirmed, and more importantly, the level of genetic modification and the search for potential of an on-target effect. And then, since the nucleus cuts upstream of the two genes here, HPG2 and HPG1, it is likely that a significant fraction of cells contain DNA deletion between the two cut sites, leaving a single hybrid gene with the HPG2 promoter fused to HPG1 gene. Whereas this deletion is expected to give rise to higher fetal hemoglobin as well, it is not clear what may be the kind of chromosomal modification that may occur at this site. Maybe, as we discussed earlier, a higher risk of copy-neutral loss of heterozygosity or terminal deletion. This, for sure, will have to be looked at carefully. One more product called OTQ923 by Novartis and Intelia, similar to the one from Editas, but based on Cas9 this time and not Cas12a. The first clinical results have been recently disclosed in December 2022. They showed that engraftment went well as for the other trial, but fetal hemoglobin production was apparently lower, as you can see here, than what was obtained with Cas12a and the Editas system. This study was interesting anyway, as it showed an important proportion of the cell with this 5 kb fragment deletion, as expected based on the presence of one CRISPR-Cas target site in each of the gamma globin promoter. Well, even so, the results were not too bad. For some non-disclosed reason, Novartis has decided not to pursue the development of this program, perhaps due to the fact that the results were somewhat disappointing compared to those of Vertex, Bluebird, and Editas. Always with the idea of directly modifying the promoter of the gamma globin gene, but also with the objective of reducing the risk of chromosomal rearrangement associated with double-strand breaks, whether at the expected location or elsewhere, Beam Therapeutics has chosen the option of using base editor. They are the first one to try this strategy in a clinical trial. They have chosen to modify the promoter of gamma globin gene by introducing mutation known to stimulate their expression, in particular by fixing the transcription factor KLF1. So far, we have only in vitro results showing the technique to work up to more than 80% of alleles in hematopoietic progenitor cells and high fetal hemoglobin production in erythroid cell. The trial was just authorized to start and is expected to include 15 subjects with severe sickle cell disease. Well, a solution that I did not talk about yet is the use of homologous recombination to directly modify the sickle mutation. I didn't speak that much about this possibility because it was thought not to work very efficiently. But anyway, Graphite Bio used the CRISPR-Cas9 and the guide RNA to generate a double-strand break adjacent to the sequence to be modified, and they use a correcting DNA template given through an AAV vector of the AAV serotype 6 that is known to enter into hematopoietic cells, so that the cellular machinery use it to repair the break by a copy and paste method. This method, they have shown it to work in human cells transplanted in human immunodeficient mice and to improve a humanized mouse model of sickle cell disease. And despite several issues, such important issue to my opinion, such as a drop in engraftment efficiency compared to what is obtained with non-treated cell, such as limited proportion of corrected hematopoietic stem cell, around 20% only, and quite large proportion of on-target indel instead of homologous recombination, Graphite-Bio was granted authorization to start a phase 1/2 clinical trial with the aim to enroll 15 sickle cell patients. This authorization was based probably on the fact that 10 to 20% chimerism in allogeneic transplantation is sufficient to reach significant improvement in sickle cell symptoms. The first patient was injected in August 2022, but unfortunately the company alerted the FDA of an unexpected adverse event. The patient in fact remained pancytopenic and needed to be transfused and treated with growth factor a few months after infusion. The sponsor decided on its own to pause the trial and said that it was focusing on the role of AAV6 in stem cell toxicity. Indeed, AAV6 is very efficient to deliver DNA for homologous directed recombination in many cell types, but the AAV genome, which is sensed by the DNA damage and innate immune response pathway, may also alter the survival and self-renewal capacity of long-term hematopoietic stem cells. This issue may be part of the explanation as to why this first patient maintained low blood cell count requiring transfusion and growth factor support. The company has thus recently decided to stop the trial. One of the last possibilities is to correct the nucleotide causing the amino acid change not by homologous recombination but by using base editor. Unfortunately, conversion of the valine into the normal glutamic acid is not possible using base editor, as there is no base editor that can convert thymine into an adenine. However, it is possible to convert thymine into cytosine. This action would convert the valine into an alanine, the presence of which is observed in hemoglobin called G. Makassar hemoglobin because it was discovered in the Makassar city of Indonesia. This hemoglobin does not polymerize and gives normal hematological parameters in both heterozygous and homozygous state so that conversion of HBS to HB Makassar in cells should give rise to cells that behave similarly to sickle cell trait. The Beam Therapeutic Company, which is already looking for base editing to create a KLF1 site in the 2-gamma globin promoter, has also a program to develop this modification, which is based on base editor, and they plan to start another clinical trial where they would try this possibility. They also obtained impressive results in vitro with more than 80% edited hematopoietic cells and a high production level of the corrected hemoglobin. We have no result, of course, for any kind of clinical trial. You may have understood now there are many options to cure sickle cell disease by gene therapy: lentiviral vector, CRISPR-Cas to disrupt BCL11A expression, CRISPR-Cas to disrupt target of BCL11A and other transcription repressor, zinc finger nuclease as well, even though they seem to be a bit less efficient than CRISPR-Cas technology, and finally the CRISPR-Cas derived base editing technology. Correction using a DNA template and homology-directed repair seem to be more difficult to reach today. In this presentation, I have shown the result for sickle cell disease, but be aware that patients with beta thalassemia are treatable by essentially identical product. This is the case for lentiviral vectors that express the beta or gamma globin chain, or for CRISPR-Cas based tool that aim to reactivate the expression of the gamma globin genes. So there have been the same kind of trial and result with beta thalassemia patient, and finally the only gene therapy product approved today is Beticel for a beta thalassemia patient in the US, which is the Bluebird Bio product and which uses exactly the same technology as that described for Lovocell. Beticel was already accepted in Europe in 2019, but due to a lack of agreement on the reimbursement terms with Germany, England, France, Italy, and Greece, Bluebird Bio, unfortunately for the patient, disengaged itself from its activity and the European market. Even though the pipeline of ex vivo project for sickle cell disease was apparently overcrowded, and it may be the impression that you have after this presentation, it now seems a little less crowded. Two candidates indeed have recently dropped out of the race, Intelia Novartis, OTQ923, and Graphite Bio Nulacell. Sangamo is now looking for a new partner for phase 3 development, but the project might be too far behind CRISPR Vertex Excessile to draw much interest. And also Bluebird Bio has decided not to support anymore the BB694 project to focus more on the development of lovo-cel. In total, there are two complementary different products, lovo-cel from Bluebird Bio and Excessile from Vertex and CRISPR Therapeutics, one based on antiviral vector and the other one on CRISPR-Cas technology. These two companies have very recently submitted their product for FDA approval for Bluebird Bio and also for EMA and UK approval for Vertex. Of course, follow-up studies over many years will be needed to confirm if these products are true one-time therapy and to what extent these two products correct both vaso-occlusive event and hemolysis. The impact of this treatment on long-term correction of the disease and on quality of life will also have to be studied and compared to that measured upon bone marrow transplantation. One of the most important issues for sure today is patient safety, and it will be important to understand long-term risk associated with these two therapies. The rate of hematologic malignancy in the HBZ206 group A patient was particularly high. Even though it was not linked to insertional mutagenesis, it will be very important to confirm that protocol modification was sufficient to reduce the risk. Also today, clearly, there is not enough experience with double-strand break technology. It will be essential to carefully follow the patient in the long term. This is why the recently developed base editing technology, such as the one developed by Therapeutic, even so there is no data yet based on single-strand break, could prove interesting, even if it seems to come a bit late compared to that of Vertex or Bluebird Bio. Only time will tell it. The competition is much welcome, thanks to Editas and Beam Therapeutics, given the setback of Beticel in Europe, for which Bluebird Bio and the institution responsible for reimbursement have not been able to agree on the price, much to the dismay of patients. So here is the take-home message. We can say that hemoglobinopathies and beta-globin gene mutation have been a tremendous stimulus for the search for gene therapy solution. As you have seen, there are a considerable number of solutions for the treatment of sickle cell disease and hemoglobin disorders in general with gene therapy. Two gene therapy products today based on very different technology are awaiting marketing approval for sickle cell patients in 2023. The progress of CRISPR-Cas technology has been meteoric, but clinical trials in this field have benefited from the experience gained in lentiviral vector trials. The next step, for sure, will be in vivo gene therapy. The number of sickle cell patients and the presence in low-income countries will not allow the majority of them to be treated by hyper-sophisticated and too expensive techniques as they are today.